So, good morning, everyone, again. Welcome to this inaugural symposium. Since I've talked already enough today, I'll be very brief. This is the first or inaugural symposium of the inauguration year, and my colleagues at CMU have done a phenomenal job of coming up with a theme that not only resonates with me personally, but I think hopefully resonate with all of you. And it sort of, to me, symbolizes what CMU is, crossing boundaries and transforming lives. And as you know, in the discussion that we just had a few minutes ago, in a conversation that we had, one of the unique strengths of CMU is its, historically, has been its boldness in going from one discipline to another discipline in the process of creating a whole intellectual domain that may not have existed. Think about areas that we take granted for now. Machine learning. Before there was a brain initiative bringing together psychology and biology, the basis for cognition, which is what I mentioned earlier as an example for learning outcomes, rather than just teaching, an emphasis on learning. These are unique aspects of CMU, and when my colleagues here mentioned to me about their desire to frame the inauguration events around the theme of crossing boundaries and transforming lives, I thought, what a wonderful opportunity. So I'm really grateful to them for thinking of this. Soon after they came up with it, the CMU co-director of the Center for Neural Basis for Cognition, Dr. Michael Tarr, who's here, he and his colleagues, of course the other co-director is from University of Pittsburgh, Peter Strick, they came up with the idea of having a symposium related to dealing with brain and research into brain. And as I mentioned a few minutes ago, this is a topic that has gathered a lot of momentum over the last decade. For example, about 10 years ago, around 2003, the National Academy of Engineering identified one of the 14 grand challenges facing humanity in the 21st century as that dealing with the human brain. And they, coming from an engineering organization, they had to put an engineering spin on it, so they called it reverse engineering the human brain. And that was one of the grand challenges. So if you go to the website, if you Google grand challenges at NAE, you will find the 14 grand challenges, and this is one of them. For the last 3 years in Washington, various congressional committees have been dealing with finding appropriate resources to fund brain research. And in fact, in the agency that I worked with, and Dr. Phil Rubin, who's in the Office of Science and Technology Policy, there was a lot of conversation about bringing together different entities that would work together in crystallizing a vision for the nation in this important area. And it's not just the US that's thinking about the importance of brain research. You may know that in Europe, there is a significant activity, and École Polytechnique Fédérale de Lausanne, EPFL in Geneva, Switzerland, recently was the recipient of a multi-institution award into brain research. And it's not just an academic exercise. It's a major day-to-day issue. And there are many different ways to look at this. More than 50% of all the Americans over the age of 85 will develop Alzheimer's. Combine that with other brain diseases, diseases that affect the human brain, Parkinson's and many other diseases. And the societal need for that is compelling. Add to that, in the last 10 years or so, the fraction of the soldiers who are returning from the two wars that the US has been engaged in, Iraq and Afghanistan, a significant fraction of them, more than 20%, 25%, have traumatic brain injury. This is an area that involves so many different angles. It pretty much involves every branch of intellectual pursuit that you can bring in. Advances in technology on the one hand, understanding human behavior, human psychology on the other hand, societal need, healthcare, cost of healthcare. It brings everything together. Plus you add to that the issue of big data, imaging, reverse engineering, circuits, electrical engineering, all kinds of fields. It's a field that's going to revolutionize this century. And these are things that we anticipate. There are a lot of things we don't anticipate, and history tells us that we are not capable of anticipating the fruits of technology, both good and bad. Let me give an example of something that we did not anticipate, good outcome. In the 1950s and 1960s, you may know that the Department of Defense started research into what we now know as GPS, or Global Positioning System. And this was purely an espionage activity during the Cold War involving satellite applications so that we can, this is for national intelligence purposes. And of course, the Department of Defense invested a lot of resources into it. Then in the 1950s, 60s, 70s, and through the 80s, the National Science Foundation put a lot of money into basic research that involves research into GPS involving mathematics, physics, solid-state physics, computer science before there was a formal department of computer science in any university in the country. And who would have known at that time that GPS would be something, a technology, that you and I will be using in all our mobile devices today? And you can't get to this auditorium if you didn't have GPS in your mobile device. And I think this is one of the greatest outcomes of funding, and that's why I was really delighted. I was part of the conversation last year, but just a few weeks ago, the president announced the BRAIN initiative, and that's going to revolutionize the scientific enterprise in the country. And also it'll impact society, not just the US, but the global society in ways in which that we cannot anticipate today. And Pittsburgh in general as a city, and CMU and University of Pittsburgh together as partnering entities have played a leading role. The Center for Neural Basis of Cognition, which is a joint effort between these two institutions, has been a leader in pioneering research in this area. And I'm really delighted that they have chosen this topic as the very first symposium of the inauguration year. And Dr. Michael Tarr, who's the co-director, and we also have Maudlene is going to be the new co-director of this center on the CMU side. They're planning on the CMU side, and it's a partnership between our two universities. And we have distinguished colleagues from other institutions, and especially I want to acknowledge my former colleague from MIT, Emery Brown, who's here, right here. And Emery is an interesting example of an individual who interfaces between technology and medicine, and with appointments in the medical school. And he's somebody who has crossed that boundary as well. In a sense, it's easier for him to interface that boundary, but he's also interfaced a more difficult boundary, which is Harvard and MIT together. So, Emery, thank you for taking the time and being here. So I very much look forward to the symposium and all the symposiums that are to follow. And I think, Michael, are you speaking next? So thank you very much and welcome to the symposium.
Thank you, President Suresh. I want to welcome everyone to the symposium, and thank you for coming on this lovely day. And I wanted to give a very warm personal welcome to the Sureshes, to both Pittsburgh and the Carnegie Mellon community. And I think one of the ways of introducing the Pittsburgh community is I found out last night that Dr. Brown actually has family in Pittsburgh and his mother grew up in Pittsburgh. And I said, that's why you're such a nice person. But it really is true. I think it's a lovely, wonderful community, both personally and academically, which is one of the reasons we all work together and love being here. So we have 5 terrific panelists. I don't want to spend a lot of time beforehand. I want to get to them, but I just wanted to mention a little more to reiterate what President Suresh said, that the BRAIN Initiative and its impact is going to be tremendous for really all of society in ways that we can't anticipate, but ways that we can't anticipate as well, because, for instance, anyone who knows someone or has themselves suffered a head injury or has had a brain disorder or is likely to get a brain disorder, there's 2 facts that stare you in the face when you think about these things. One of the facts is that we don't really have a good way or definitive way of diagnosing any of these things. The second thing is that even if you can diagnose them, we usually don't have a good definitive way of treating or curing any of these things. So the brain and all of the things that can happen in the brain are tremendous mysteries to us still, and we're working hard to solve them, and Pittsburgh is a great place for doing that, but the challenges are still set before us. So the importance and the impact of the BRAIN Initiative is very, very clear. We have limited knowledge about how the brain works, and it's only through the sort of science we're going to hear about today and that we do in Pittsburgh that we're going to be able to ultimately understand something as complex and as amazing as the human brain. So I wanted to introduce our panelists. Dr. Philip Rubin he's the Principal Assistant Director for Science at the White House Office of Science and Technology, and an old acquaintance from many years ago in New Haven. Dr. Tom Mitchell, who's the Fredkin University Professor and Chair of the Machine Learning Department at Carnegie Mellon. Dr. Nathan Urban, who is the Schwartz Distinguished Professor and Head of the Department of Biological Sciences at Carnegie Mellon. And Dr. Marlene Berman, who's a Professor of Psychology and in the Dietrich College of Humanities and Social Sciences at Carnegie Mellon, and as President Suresh mentioned, the incoming co-director of the Center for the Neural Basis of Cognition. And Dr. Emery Brown, who is a Zappel Professor of Anesthesia, Harvard Medical School, and the Professor of Computational Neuroscience in the Department of Brain and Cognitive Science and Professor of Health Sciences and Technology at MIT. And as I said, has roots in Pittsburgh. So finally, before we start, I wanted to mention, we want to ensure we have enough time to hear everyone. So I'd like to hold questions for a panel discussion we're going to have at the end, but during the entire series of talks, Jen and Peter, there they are going to be passing out cards which you can write questions onto, and they will collect them and then give them to me, and I will pick through some of the questions and we'll ask the panelists, although I may have some surprise questions of my own for the panelists as well. So without further ado, we'll start with Phil. Thank you.
Good morning, everybody. First of all, I'd like to thank President Suresh, Mike Tarr, and all of Carnegie Mellon University. I'm humbled to have been invited to this important and wonderful event. Time is very brief. I'm going to provide a very broad overview, and I'm going to do it from more of a policy perspective. Then we're going to be getting into some incredible talks on the details of the science. So mine will be, in a way, a little more content-free, and will give you a perspective from inside the Beltway, but also building on over a decade of planning across universities and agencies. So I'm going to be talking about what we broadly call the White House Neuroscience Initiative. I work at something called the Office of Science and Technology Policy, and most of you have never heard that. That's in the Executive Office of the President. And I'll give you a sense of what OSTP, there are a lot of acronyms in government, so I'll try to spell them out, but then go with the acronym after I use it the first time. OSTP coordinates the development and implementation of the administration's domestic and international science and technology policies, programs, and with the Office of Management and Budget, OMB, oversees development of department and agency budgets in support of the President's science and technology goals, notably strengthening the economy and creating jobs, improving health care, addressing energy and environmental challenges, advancing national and homeland security, maintaining U.S. strength and basic science and space exploration, and lifting national performance in STEM education. Today I'm going to be focusing, as I mentioned, on the White House Neuroscience Initiative, which is a very, very broad initiative, and I'm going to give you a sense of that. What are the goals of this initiative? They are to develop a strategy towards significant transformative discoveries in fundamental and translational neuroscience and cognitive science through coordinated federal efforts and synergy across outside stakeholders. At OSTP, we try to make sure this coordinates with the administration's priorities and with other initiatives and activities. Why are we doing this? We've heard from Dr. Suresh in part about the societal and human costs, and he gave an example. Here are some other examples of the economic, human costs and challenges that we face as a nation, and they're broad. I just picked a few out there. Neurological disorders affect more than 50 million Americans every year, cost more than $500 billion a year to treat. It's been predicted that delaying the onset of Alzheimer's disease by just 5 years could save $50 billion in annual U.S. health costs. And there are a couple other examples. So this is a profound economic, human, and societal problem. And you'll see some of the other challenges that we have to face. So because of that, the administration from the start, from the beginning of the administration, has been broadly engaged in the area of neuroscience. And you can see from this slide, if you're able to see it, I hope you can, but I'll go through it and I'm going to unpack it. Some of the examples of the activities that we're engaged in currently and that we've been in before. I've listed just a few of them up here. This is a broad initiative. Borrowing other people's words, this goes from neurons to neighborhoods, from molecules to mind. And it spans many different activities, including those listed above. I'm going to briefly touch on each of those. I'm going to try to concentrate a little bit on the BRAIN Initiative. First is the National Plan to Address Alzheimer's Disease. This has been going on since the start of the administration. And in the main, it's led by the National Institute on Aging at the National Institutes of Health, but engages many of the agencies of government and has international cooperation. And you can see, I hope, again, from the slide, some of the key points. This past year, there was an Alzheimer's Research Summit called Path to Treatment and Prevention, and it had developed important recommendations. If you'd like to find out more about this, you can go to www.alzheimers.gov or just Google Alzheimer's NAPA, National Action Plan, and you'll get some of the details of the recommendations. More acronyms. I'm going to be talking to you in a moment about something that emerged, and I was asked to come down to Washington to take a leave from what I was doing in the past, and to create a group under the auspices of something called the National Science and Technology Council. It is run by OSTP, and it has a number of committees in different areas. I've listed some of them, including environmental, natural resources and sustainability, homeland and national security, STEM education, science and technology. I've had the pleasure of co-chairing with Dr. Suresh and Dr. Francis Collins, the Committee on Science, and I continue to do that. The next thing that I'm going to be talking about, and the reason I put this acronym up there, is that when I came down, it was because of some congressional language in part and planning activities requesting that our office, working with the agencies of the government, create an interagency working group on neuroscience. The mission of this interagency working group is to coordinate activities in neuroscience research across the federal government and focus on the fundamental understanding of learning, brain development, and plasticity, and applications to brain health and recovery. It started out by identifying key research areas and then defining concrete actions that can be taken, including things like the BRAIN Initiative and others that we'll be rolling out. This is a very broad activity. It's much broader than the Brain Initiative. The Brain Initiative right now partners 3 agencies. So this involves over 20 entities of the government, and the print is very small. I was really putting this up for those to see and those who can't see, to show that almost every agency of the government is engaged and has what we call equities, interest in neuroscience. Everything ranging from the National Science Foundation, a co-chair, National Institutes of Health, through the Environmental Protection Agency, NASA, Department of Justice, Office of Director of National Intelligence, Department of Education. So when you create an interagency group through the NSTC, usually you're bringing all the agencies of government. Some participate more than others, depending upon their degree of interest. There's a lot of interest across most of these agencies. Our progress to date is that we delivered an interim report, and that we're finalizing the final recommendations. The NSTC tends to be inside the government, and its activities generally are not public. Its activities are not public. We are very interested in making a portion of our work available to the public, so we're working very hard on creating a public report. The reason we can't talk about everything is because some things, particularly agency things like at the NIH and NSF, might be talking about programs that are in the planning stage, and it would be unethical, illegal, and inappropriate to be talking or providing anybody with information about stuff that's called deliberative or pre-deliberative. And so those kind of things, the agencies are very sensitive about, and so are we. So hopefully, we hope in the mid-November timeframe, there'll be the public thing. I can give you some hints, because I've talked publicly about this, that the recommendations seem to be focusing on understanding and treating brain disease, disorders, and traumas, understanding and optimizing interactions between the environment and the brain across the lifespan, understanding and applying the brain's information processing capabilities, and enhancing communication across federal agencies and with the public. We're already doing the things I'm talking about. I can tell you about because we're already doing and have talked about them publicly. Our next step is to get this, as I mentioned, to get this out publicly and to probably recharter this working group. There's also in August the 31st of 2012, the President issued an executive order on improving access to mental health services for veterans, service members, and their families. The reason I wanted to mention this, aside from it being important, it shows some of the breadth of the neuroscience interest, but a part of it was a national research plan, particularly around some things that Mike Tarr mentioned, which is diagnosis, treatment of concussion, both for our military, but also for others who have suffered from traumatic brain injury. We're also looking at suicidality and PTSD, post-traumatic stress disorder. The Brain Initiative has grabbed the most attention. It's actually, as Dr. Francis Collins points out, the NIH investment, which is not the biggest of the agency investment in 2014, is about $40 million, and their annual investment in neuroscience is $5.5 billion. So it's actually a very small investment, but through partnerships, as you'll see in a moment, across the agencies and public-private partnerships, this has grown into a much larger effort. However, what really sparked the interest was the President mentioned this in his State of the Union address. We're always delighted that language like that gets in there. We can never predict what will get in there. And so we were very happy to see that, but the second that happens, you know that you also have to manage expectations and try to get focus on things. The other thing that happened is a story appeared, actually the evening before, on the front page of the New York Times on the top. Anytime science is in the newspaper, no matter how, I wouldn't say inaccurate, but perhaps well, many of us are familiar with newspaper stories. If they get your name spelled right, that's great. But it's a rarity, and we love the press. However, you really then have to manage expectations, because there were quotes in the stories about things that just weren't going to happen, like $500 million and billions of dollars. There's very little money, and whatever money is out there, we need to use carefully. We need to think about what's going on, and it's not a top-down activity. This is not about us saying what's going on. It's, as Dr. Suresh said in his earlier speech, about listening to what people including average citizens, through scientists, through other stakeholders, advocates, what they think is important, and trying to assess what is it we can do. We do not have a lot of levers, and spending money is certainly not, it would be the last thing that we focus on. It's got to be about critical importance to the nation, and it's got to be about excitement and vision. If that's there, then the money can come. And if somehow we can work with private corporations, foundations, who are willing to help leverage the very limited money of the federal government, that's exciting. And in part, that's why we were interested in this. And you'll see that in a moment. But the three key partners in this initiative are federal agencies the beginning, and they are DARPA, the NIH and NSF, and I'll show you them in a moment. You're going to be hearing more about the BRAIN Initiative, so I'm not going to focus on the details. I did want to mention, though, why we're doing this. And part of it is about crossing boundaries, as the session's named, trying to work across the sciences and integrate the findings, as Dr. Suresh said, across many different areas of activities, as I said, from neuron to neighborhood, physical, physiological, genetic, cognitive, behavioral, to support curiosity-driven science, to create new tools, technologies, and to particularly improve data management. To do this, we're going to need time, and we're going to devote time. We consider this a long-term investment. DARPA is one of the partners. I invite you to go to their website to learn details about what they're doing. They're building on existing programs, including the Revolutionizing Prosthetics. They have two other major interests. Well, they have several. One is in the data aspects, and the third is they pushed us very hard. The director of DARPA's a woman named Arthi Prabhakar, and she said, we're not going to do this if we don't think about the ethical, legal, and societal impacts. And so we'll get to that in a moment. Next partner that you've heard a lot about is the NIH. Recently, a working group of their advisory committee to their director provided a set of recommendations and will deliver a final report in June on a set of directions that they hope that the NIH will focus on. They range from creating a census of cell types and creating structural brain maps to broader attempts to look at the circuitry underlying thought, behavior, and action. And again, just go to Google NIH Brain Initiative, and you can download it's a 58-page PDF file that's freely available that provides the details. The National Science Foundation has actually led the way in this area. Before the Brain Initiative came out in March of last year, they issued a Dear Colleague letter across all the directorates. The acronym for it is AIR, NCS, AIR, -NCS, and it stood for Advancing Innovative Research in Neuroscience and Cognitive Science. And it was a message from the National Science Foundation that they believed that all of the directorates of the National Science Foundation could play an important role in this activity. More recently, they've put additional information out, and one can expect future announcements. It's kind of clear from the information that's come out of recent workshops and recent announcement that there's a set of key areas: identification of neural signatures that predict complex behavior, development of computational models, establishment of quantitative and predictive theories of brain function, and support for unprecedented levels of data sharing. So that issue of data sharing and the sharing of resources cuts across three agencies. Time is short, so I'm going to go quickly through the rest. We also had partners, not from the federal government, and they'd include the Allen Brain Institute, $60 million annually, Howard Hughes Medical Institute, $30 million annual contribution, Kavli Foundation, which really should be credited for helping to get this effort started by getting the other foundations together and trying to get them together with agencies in the federal government, are contributing $4 million annually for a decade. And the Salk Institute, we're reaching out to other foundation partners and also to corporations. As I mentioned, the ethical aspects of this kind of research need to be kept in mind. They need to be really a primary thing. So from the beginning, we decided that it would be important to engage the President's Commission on Bioethics, in particular because of their role of reaching out to the public in the activities that they do. That commission is chaired by Dr. Amy Gutman, president of University of Pennsylvania. And you can just go to www.bioethics.gov to see the charge that the president provided them. Moving away from the BRAIN Initiative onto other topics, I did want to mention that neuroscience is very broad. The president has begun a national conversation on mental health. In addition to the work for the Veterans Administration that's about research, this national conversation, which will be focused, I think, in the main at the Department of Veterans Affairs, is about reducing a stigma related to people who need or might consider getting treatment. And that is really a profound challenge, particularly with increases in suicide. It's important to find ways to get people who need to reach out for assistance of encouraging them. So we were delighted to see another event in the East Room. So the particular focused activities related to that include, in the current year, expanding mental health coverage, supporting young people through additional funds to help teachers and adults recognize signs of mental illness, improving access, as I mentioned in the other executive order, for veterans, and supporting research, most of it focused at the institutes of the NIH. So in summary, I'd like to close by saying that broadly speaking, the goal of the White House Neuroscience Initiative is to coordinate the federal efforts and help build public-private partnerships that accelerate our understanding of the human mind and have the potential to help uncover new ways to treat and prevent and cure brain disorders like Alzheimer's disease, schizophrenia, autism, epilepsy, and traumatic brain injury. The BRAIN Initiative builds on the President's call for historic investments in research and development to fuel innovation, job creation, and economic growth. The initiative fosters cutting-edge and interdisciplinary research needed to understand how brain function is linked to behavior and learning and the mechanisms of brain disease. And I don't think anything could be better to summarize our interest than the title for this particular symposium, Crossing Boundaries and Transforming Lives. That's what our goal is. In a nation like the United States, those boundaries are fairly broad. And so we are trying to work across all sectors of American society to help people through the limited efforts that we can make of the federal government, and to also recognize that the talent and expertise exists locally in the universities, towns, cities around the country. Thank you.
Well, Mike Tarr asked us to think about how the BRAIN Initiative, and actually a collection of initiatives that Dr. Rubin just discussed, will affect us at CMU, and perhaps more importantly, how CMU could affect this collection of BRAIN initiatives. I want to think about that together from the point of view of a computer scientist, which is what I am, And when I was preparing my notes here, it struck me that, in fact, there are two very different synergies between the brain initiatives and the field of computer science. One of them has to do with artificial intelligence. So computer science, artificial intelligence, is the field that looks at the question, how do we program computers to do those kind of things that we label as intelligent in people? How do we get computers to understand natural language, to see things, to do computer vision, to do problem solving, complex problem solving, to hold conversations? And that area is an area that CMU has long been a pioneer in. In fact, when I was a graduate student elsewhere at Stanford, I was well aware of the work that was going on here at CMU by some of the pioneers of the field, like Herb Simon, Alan Newell, and others. And that quest for understanding intelligent processes within the field of AI has to do with trying to figure out what are the algorithms, what are the sub-processes. And the BRAIN initiative, one of the key efforts there, is try to understand from brain data how it is that the brain is conducting those same processes. So one of the areas that's an obvious area for synergy is for these two fields that have been fairly distinct historically, artificial intelligence and cognitive neuroscience, to come closer and closer together. One of my favorite examples today is actually some work done by one of our faculty in the psychology department with a joint appointment in computer science, John Anderson, who has a computer program that solves algebra problems. You give it a simple, remember 3x plus 7 equals 10, what's x, kind of problem. It solves that step by step, and as a side effect of solving it, it predicts the neural activity that one will see in the brain if you give the same algebra problem to a person in an fMRI scanner. So I think John's work is an early example of the kind of synergy that I hope we see much more of, where we have an AI program solving the same problem that we give as stimulus to a person in a scanner and try to see that correspondence and use one to learn about the other. So that's one of the two areas that struck me. The second area that I think is the one that I want to actually spend the rest of our time talking about is that the BRAIN initiative, as Philip mentioned, is going to lead to big data. It's going to lead to the largest collection and the most diverse collection of data that we've ever had on the brain. And so for us, that offers a big opportunity to develop computer methods to help semi-automate this new big data empirical science. I happen to be the chair of the machine learning department, and machine learning is the field that develops the underlying algorithms that allow us to look at these large collections of data to discover regularities that are just too subtle for people to pull out. So let me ground that by talking a little bit about some of our own research that I've been doing for the last 5 to 10 years with a collection of very good-looking people. We've been putting people in scanners, and we're interested in language processing in the brain. That's the focus of our work. To get started, we thought, well, let's just look at single-word language. Let's put people in the scanner and show them words. So, in fact, in this case, here is a fMRI image. Looks like 4, but it's actually 4 slices of a 3-dimensional brain image. When somebody was shown this stimulus up here in the corner, the word bottle appearing underneath a line drawing of a bottle. And the way to read this, as the thermometer on the right shows, is red means high neural activity, dark blue means below average neural activity, and what you see there is the top of the brain, or the top of that picture is the back of the brain back here. And the bottom of the picture is the front of the brain. Okay? And so you can see there's splotchy red activity in this person's brain when they think about the word bottle. Okay, so now, interesting question. Can we study the question of what are the patterns of neural activity that actually constitute the representation of the meaning of bottle. For example, is this any different than it would be if I showed some other word? Well, here's, in fact, the brain activity averaged over 60 different words. If you squint, you might see a few differences with bottle. But so our very first question was, can we study and can we use brain imaging to get a grasp on how brains use neural activity to represent the meaning of the ideas that you're thinking about? So to begin, we leaned back on a machine learning method. We said, well, why don't we train a classifier, a computer program? We will give it training examples of the form, here's a brain image, and this person is thinking of bottle. Here's a different brain image, this person, and the same person is now thinking of house. Here's another one where they're thinking of bottle. We give it training examples like that, and the same kinds of algorithms that learn to detect spam, if you show here's an email that's spam and here's one that's not, similar algorithms can look at brain images labeled and learn to classify the subtle patterns of neural activity that distinguish thinking about, say, bottle from house. So we tried this, and in fact, it works. So this is one interesting way to think in very concrete terms about the way that machine learning algorithms can play a role in cognitive neuroscience. This classifier that we've trained becomes a virtual sensor, not of the brain activity, but of the information content in the person's head. It becomes a virtual sensor of the mental state of the person. The brain image just shows you what the neural activity is, but we can now tell you what the mental state is, or at least this trained classifier can. Why is that important? Because now we can start using that virtual sensor that we trained to ask very interesting questions. What about your brain and mine? Does your brain represent bottle the same way my brain does? How could we answer that? Well, here's the obvious way to answer it. Once we have that classifier trained from data from my brain, we can put you in the scanner and we can see whether that same classifier, which has been trained on my patterns of neural activity, can be used to decode yours. We tried it. It works. One of the things that we discovered as a side effect of this is that even though we're all very different, we are remarkably similar in terms of the patterns of neural activity that represent the meaning of very diverse words. And so your brain is more similar to mine, and your brain probably looks like that when you think about bottle too. But the point, the scientific point here is this is a game changer because it lets us shift from the raw data, which shows us the neural activity, to another level where we're talking about mental states and ideas that people are thinking about. Another experiment, we did the same kind of thing. We said, somebody asked, well, what if you have somebody who's bilingual and they see the word bottle in English, but then they see it later in Portuguese? Will it be the same or different? We did the same thing. We trained the classifier on English data, English word stimulus data, and then we tested whether it could decode the neural activity when somebody read the word instead in Portuguese. It worked fine, just as well. It doesn't actually matter what the language is. Okay, so point one, this gives a way of essentially training systems that become virtual sensors of information content in the brain. Now we can form a whole scientific research agenda on top of that. But I want to move on to a different point, which is somehow having a classifier that tells when you're thinking about bottle and a different one that tells when you're thinking about hammer is not really a theory of how the brain represents things. It's a list of how the brain represents bottle and hammer and so forth. So what would it mean to instead seek a theory of neural representations of meaning? Well, everybody knows that theories they're logical systems that make testable predictions, a phenomena that they have not yet been exposed to. So one thing it might mean, or part of what it would mean, to have a theory of neural representations of meaning, is that we'd have some kind of a predictive theory or predictive model where we could input any noun, and that theory would tell us, make a prediction of what would be the neural activity when a person thinks about that noun. So we worked on that for a while, and we came up with one. It's not perfect, but it's pretty good. You can give this, and again, it relies on machine learning algorithms to train the model in a way where the scientist, that's us, first constructs the form of the model. That's our contribution. But the model leaves open 500,000 parameters whose values we don't want to commit to, and that get trained by training data of the form, well, here's a word and here's a brain image. But once this model is trained, it can then extrapolate to make predictions for any noun. And here's how it works. We input a word, suppose the word is celery. The model first represents the meaning of that word by how often celery occurs with 25 different verbs when you look at 1 trillion words of text from the internet. And then it uses those features to predict with a linear regression the activity in each of 20,000 different locations in the brain that we can observe with fMRI. So I don't want to go into too much detail, but for example, for celery, here are the verbs. Celery occurs a lot with eat, not very much with ride, whereas airplane occurs a lot with ride. And so when the model is done, it's able to make predictions for words that it has not yet been trained on. They're not perfect, but if you compare the predicted to the observed, you can see they're similar. Okay, so I want to end with one important point, which is what I think is actually the biggest. So 2 points. One, in a sentence, what we've seen here is that by integrating and inventing new machine learning methods to go with the kinds of neural data that we can see, we can open up new ways of modeling and studying the neural data. But the big opportunity going forward, I think, is actually to get out of the mode, the paradigm that the field is in right now, which I would call the single experiment paradigm. Single experiment science is what we just saw, what I just showed you. We collected some word data, we trained a model, we actually wrote a paper, and now we're writing another paper on a different set of data. But what we want to do, the big opportunity, now that the Brain Initiative is going to give us this flood of more data than we've ever seen, and diverse data. We'll have data where people are looking at words, we'll have data where people are looking at sentences, we'll have data where people are reading stories, we'll have other data where they look at letter strings that aren't really words. And the big opportunity is actually to train much more subtle and comprehensive models of things like language comprehension in the brain by using not just one experiment's worth of data, but thousands of experiments worth of data from different people, from different labs, some of which are experiments with single words, some of which are with sentences, other of which are stories. And if you think of just that case study, this little story in the context of language processing, you can repeat the same kind of story for vision, for recovery from stroke, for many, many different things. So I think if you get one point out of this talk, it's the big opportunity going forward is innovation at the interface of data analysis, machine learning modeling methods, and consumption of this very diverse, high-volume collection of experimental data we're about to get our hands on. Thanks.
So, thanks very much. today I want to tell a brief story and that makes a couple of different connections to the BRAIN initiative, and I want to start with some of those connections. I think two things that are critical components of this. One is along the lines of what Tom mentioned, which is the attempt to consolidate data, information, results from across many different labs. And the second is to have a good connection between our understanding of the human brain and our understanding of the brain of model systems, for example, mouse models of disease. So I want to tell a story that touches on these two themes. But the story really starts with a broken faucet. A few weeks ago, I had a broken faucet. I started to try and take it apart, and in the process of doing that, I wanted to know what I should find when I took that faucet apart. And so in order to do this, I went online and I searched for a diagram of that faucet, and I came up with something akin to this. Now, this is not a faucet. This is, I believe, a motorcycle engine, but it still makes the point, and a point that I could better make with a laser pointer, but let me see if I can. The point is that in thinking about the analysis or repair of any, here we are, thank you. The analysis, the understanding or repair of any device, be it a motorcycle engine or a brain, a couple of things are critical. One is we need to know something about the parts. In this case, this is a sort of, I think they call these exploded diagrams of a motorcycle engine. And it's important to know about the parts because you want to know about their properties. You want to know when you take this thing apart whether this gear is as it should be or not. So you want to have specifications or specs as to what those parts should be. You also even want to know a simpler thing. There are two parts up here. Are these two copies of the same part, or are they two different parts? Do we need to order something different, two things if those break, or can we order one thing twice? And I think for me, this is a very useful analogy for thinking about or trying to understand and eventually repair the brain. So the parts of a brain are neurons, the individual cells that make up the brain, and the basic function of a neuron, of every single neuron, there are actually two neurons shown here, but the basic function of the neuron is to perform an analog to digital conversion. It takes a signal, such as this one, that comes in, which is an analog signal, just random fluctuation in this case, that we actually put into a particular neuron, and it generates as its output a train of events, these discrete individual events called action potentials or spikes. These are highly reproducible, and so you can actually sort of think about this as in the same way as a binary string, and then do all kinds of interesting things by calculating, for example, the amount of information that's available in this string. Now, so all neurons function effectively like this, but there are lots of different kinds of neurons. This is an example of a bunch of neurons based on their anatomical type, and anatomy is interesting and beautiful, but for us, we want to know more about their functional or physiological type. And so one thing that we've been doing is to try and, along the lines that Tom mentioned, sort of build ways of collecting data about functional or physiological types of many different neurons. A student of mine who just recently defended his PhD thesis, Srijoy Tripathi, did a lot of this work to collect from the existing literature lots of information, lots of data about different types of neurons, data that's been collected since scientific papers started appearing as PDFs, which is not decades ago, but at least, in the mid-90s or so, to collect this kind of data, put it into a format so that many, many people could use it, created a web interface to do this. And so now we can start looking not just at experiments that come from my lab, but across experiments that come from dozens, hundreds of labs to look at the properties of these neurons and to build up this database to have a richer and richer data set that gets incorporated into it is one of our long-term goals. But we've also been interested in a separate idea, not that there are neurons of individual types, but that in some ways the brain is built using manufacturing processes, if you will, that predate the invention of interchangeable parts. Many of you may remember that I believe Eli Whitney is credited with the invention of interchangeable parts, the idea being that if you make 100 screws, those 100 screws don't go into 100 different cotton gins or whatever piece of machinery, but rather that any one of those screws can be used in any one of those cotton gins. It turns out that we think that brains are made up not of interchangeable parts, but that individual neurons, the parts of the brain, are more like snowflakes. in that individual neurons, while they may be constructed in such a way based on certain principles, the symmetry that's evident in the construction of snowflakes, but individual neurons differ one to the next in ways that are actually useful and interesting and may actually convey certain functional advantages. We think this is an important difference between the way brains compute and the way machines compute. One of the ways in which we've tried to capture this diversity, this heterogeneity, is by building models that incorporate this. And the models here, the details aren't important. The fundamental thing that I want to say about these models is that they are stochastic, as indicated by the pair of dice here. So that neurons, as wonderful as they are highly unreliable devices. And so just as an example, if you take a neuron, and this is sort of the same kind of data that I showed previously, you have a single input that you provide to this neuron. On one particular occasion when you provide this input, it generates this train of spikes or this train of action potentials. But if you do this a bunch of times now, you can actually see that this particular action potential, shown here, only happens about 20% of the time. And so this fundamentally stochastic nature of the output of neurons is something that we've been very interested in from a basic science perspective. Now, it turns out that a few years ago, Marlene Berman, who will speak next, became interested in the stochastic nature of brains at a very different level and for very different reasons. I'll skip that. Her observation was that if you look at the brains of autistic individuals, you look at the whole brain or brain areas responses to stimuli. So we've gone from single neurons responses to stimuli to now whole brain areas responses to stimuli. The brains of autistic patients show lower levels of reliability. The response to simple sensory stimuli is less reliable. The brain response to simple sensory stimuli is less reliable in the brains of autistic individuals. So to us, this is a fascinating and highly intriguing example where we might be able to relate something that we see at a cellular, individual cell level or small groups of neuron level, where we can understand mechanisms, or we can understand what proteins, what channels, what neurochemicals underlie this unreliability, and to try and now connect it to this observation from human subjects. And so for us, the way to try and bridge that gap is to look at animal models of disease, in this case, animal models of autism. Now, I could talk for a while about what makes a mouse model of autism. How do you know your mouse is autistic? And I think that there's a lot of controversy about that topic. But there are, it turns out that there's a foundation called the Simons Foundation, which has been very interested in promoting and funding research in the area of autism. And one of the things that they've done is they've done some of this work to identify genetic models where particular genes have been knocked out or eliminated, where the mice then show behavioral phenotypes that are similar to those seen in autistic patients. And so this is just a summary. And so with a new grant from the Simons Foundation, we've been able to obtain some of these mice. And so now we can ask at a cellular level whether the brains of these mice, whether the neurons in these mice show alteration in their responses, in the reliability of their responses to stimuli, and then with the hopes to try and understand how it is that those mechanisms might play out in the case of human individuals with autism. I think I'll leave it there. Thanks very much.
It's my privilege to be part of this esteemed panel this morning and to join together to welcome Dr. and Mrs. Suresh to Pittsburgh. The question addressed in my lab is how the input received at the retina, which is essentially the intensity of light and the frequency of light, is transformed into meaningful percepts. We merely open our eyes and instantly have available to us information about the color, the depth, the form, and the motion of a stimulus such as this fly. On the one hand, we know a tremendous amount about the properties of the eye and the structure and organization of the brain. On the other hand, we know about the extraordinary feats of human perception as evidenced in our ability to recognize a whole set of images as flies, even though they differ in their color, their orientation, their form, potentially even in their geometry, and even when they appear in entirely unfamiliar contexts. The key question is how this richness of visual behavior emerges with such precision and rapidity out of the underlying neural system. In today's talk, I'm going to illustrate this issue in the context of perhaps the most taxing task confronted by the human visual system, and this concerns the ability to recognize individual faces. This is a particularly taxing task because, as you see from these images from the Warhol collection downtown, faces all have two eyes, a nose, and a mouth in the same spatial configuration. The difficulty also arises because a face conveys a host of information about the age, gender, identity, etc. of the individual. As such, and notwithstanding this complexity, human face recognition proceeds accurately and with seemingly little effort. As such, face processing is a useful domain to explore the underlying neural basis of behavior. I'm going to demonstrate today 3 approaches that we can use to understand the neural basis of face recognition. I'm first going to map out for you the regions of cortex that play a role in this task, and then I'm going to delve into these regions to understand what information is coded in these areas. Finally, I'm going to tell you what happens when the system breaks down, as will be evident in a whole host of conditions, including in autism and in Alzheimer's disease. Let me start off by mapping out the regions of the brain that are involved in this process. Participants lie in the bore of a functional magnetic resonance machine. We have a wonderful magnet here on the campus. And we project into the bore of the magnet images of faces, objects, and buildings, and explore what neural populations become activated in response to these images. On this diagram, we see a graphically inflated brain. This illustration here is of the inferior or bottom surface of the brain. We've stripped away the skull and all the other structures so that you can see the inferior surface. And here is the lateral or the side surface of the right hemisphere. The different color patches indicate which regions are selectively activated. For example, red regions by faces, green regions by buildings, and blue regions by objects. And I particularly draw your attention to the fact that there is a multiplicity of nodes activated selectively in response to faces. We can also begin to explore the developmental emergence of the organization of cortex, and we can show that adolescents ages 11 to 14 are already evincing a mature neural profile, although children age 5 to 8 who are not entirely accurate in face recognition correspondingly show a rather immature neural profile with pretty much no face-selective regions identifiable. This prolonged developmental trajectory is likely a result of the fact that face recognition is so difficult, and forming this distributed, precise neural network requires a long time to emerge. Having demonstrated that there are these multiple nodes, the question is what information is coded in these nodes. And we don't want to simply illustrate that these areas are able to differentiate faces from objects. We want to be able to demonstrate that these areas can identify individual faces. And so in this next study, we brought individuals to the lab trained them to identify individual 1, 2, 3, and 4. There are a whole set of different images for each individual, and they simply had to discriminate facial identity, which is a rather difficult task given that these faces are fairly similar to each other. They then came to the magnet over a number of occasions, and inside the bore of the magnet saw one of the faces, had to decide if it was person 1, 2, 3, or 4. A little bit of time passed, another face came up, and they did this over and over again, and we gathered multiple measurements over these multiple visits to the magnet. Unlike the image that I've shown you previously of the surface of the brain, I'm now going to show you that when you cut deep into the brain, and the eyes are here in the front, and this is the back of the brain, we can again see multiple regions of cortex that have a signal that contains sufficient precision to support above chance discrimination. These regions can make a four-way discrimination across the individual faces. If it is really the case that we can understand the information representation in these areas, we might be able to computationally analyze and model the neural signal in these areas and use the signal to uncover the particular signature for each individual face. If we truly understand the representation or the neural code, we should be able to use this code to translate the neural signal and reconstruct the image that was presented to the participant. We're very much in the midst of these experiments, so I'm just going to show you some early data in which we can read out the face code. Here are stimuli that are shown to the participants, and as you see, they're extremely similar, stripped away with no identifying characteristics about hair or any other facial cues. And we show that in these key areas of the brain, we can use the neural code to reconstruct the stimulus that was shown to the participant. Now, the first piece of good news is that if we are able to reconstruct stimuli that actually look like faces. The second piece of useful information is that the faces are not all identical to each other. Now, although we are not able to reproduce a faithful and veridical reconstruction of the face, you will note that the upper part here is a rather good rendition of the stimulus. And this is consistent with the well-known fact that humans look longer at the upper part than the lower part of faces, and more informational salience is conveyed by the upper part of the face. The last part of my talk will be an illustration of the consequences of breakdown to this finely tuned neural network. In the last few years, we've had the unique opportunity to examine the brains of individuals who suffer from a disorder known as face blindness. The technical term is congenital prosopagnosia. For all intents and purposes, these individuals are normal. They have no obvious brain abnormality. They identify faces by using cues such as voice or clothing. They have never seemed to acquire mastery over the seemingly effortless process of face recognition. If you show individuals with prosopagnosia faces such as these, where control participants, the majority of control participants can identify the majority of these faces, we see that these prosopagnosics, in fact, can identify very few of them. The disorder can be so severe as to affect the recognition of even close family members. And I share with you an anecdote from one of our participants who says that when he goes to pick up his daughter at daycare, if the teacher changed her clothing during the course of the day, he's no longer able to identify his daughter and has to wait for her to be brought to him. From a scientific point of view, the intriguing question is what is going on in the brains of these individuals? We started off by mapping the face selective regions, and this depiction should now be familiar to you, in which we see the multiplicity of face selective nodes in the control participants And to our surprise, we were able to uncover these phase-selective nodes in the congenital population as well in the posterior parts of the brain. This led us to entertain an entirely different hypothesis, and that is that the impairment likely arises from the propagation of the signals between the nodes rather than from the nodes themselves. And fortunately, we've been able to take advantage of a new technology to pursue this question. Prior to this, we would only be able to examine the white matter tracts on autopsy or post-mortem. But this new technology, in which we can measure the movement of water molecules or the diffusion of water molecules, allows us to uncover the white matter tracts. For just a quick explanation, if you look at diffusion of water in, say, cerebrospinal fluid, it diffuses randomly in all directions, spherically, in an isotropic fashion. Whereas when water is constrained in a tract, such as in the corpus callosum, it can only flow along a principal direction, and we can use this gradient information heuristically to uncover the existence of these fiber tracts. We were able in control participants to uncover fiber tracts that project through this distributed neural network. There are two major tracts, the ILF and the IFOF, and much to our good fortune, we could show that these tracts are structurally compromised in the individuals with congenital prosopagnosia. As you can see in this individual, we were barely able to uncover any of the tracts, perhaps just a few little threads in the left hemisphere. We see this reduction in the tracts across an entire group of congenital prosopagnosics relative to their control individuals. And intriguingly, to the extent that these tracts are compromised, this is correlated with the severity of the face recognition ability, attesting to the functional value of these tracts. This last goal has been to begin to elucidate the nature of the impairments in the brain, so as to both be able to understand these impairments across a range of different disorders, such as autism and Alzheimer's disease as well, and begin to develop more refined and targeted intervention procedures. So let me wrap up here by saying that the kind of science that can be done at CMU and in Pittsburgh more generally employs the stated goals of the BRAIN initiative. We cooperate across fields of research. We link neuronal activity to behavior. We use innovative tools and techniques and integrate theory and computation with experiment. And in the course of executing this science, we're crossing the boundaries and impacting people's lives. Thank you.
So I just want to thank Michael for inviting me. Thank Subra for those kind words. So I'm going to not tell you about this, but tell you about this. All right. So I'm going to give you a little insight into what anesthesia is all about. Right? So, and I'm going to make it very practical. I'm going to leave you with an understanding of something which is probably the most commonly used anesthetic drug, which is propofol. And I want to give you a sense of how does it work? Why does it make you unconscious? Just this very basic question. I'm going to tell you the answer right now, because talking about anesthesia at this time of day has potential soporific potential. All right? So it creates a hum in your brain, a hum which is about of 10 hertz. And it also causes your cortex to fragment, all right? And it's going to inactivate your arousal system. So I just want to show you that, all right? And then I'm going to do this like Snoopy. You know, when he's writing a novel, he says, I'll tie it all together in the last chapter. So I'll tie it all together in the last chapter, all right? So first, what is general anesthesia? So it's this drug-induced reversible state, and it consists of being unconscious. You're not supposed to remember. It's nice if it doesn't hurt. It's convenient for the surgeons if you're not moving around while they're operating. And this part here, stability and control of the physiologic systems. Because you can take all four of these right here, and they're synonymous with death. Right? And on that long list of things that's not cool, that's not cool. all right? But we actually spend a lot of time sort of maintaining this, the physiologic stability. This is something which is said, you know, it's not, we don't understand how the drugs work. So I want to show you that that's not really the case. So how many of you have had anesthesia? Oh, I got a little anesthesia club here. All right, how many people have seen other people get anesthesia? Okay, I'm going to fix that for the rest of you then. All right, so this is a gentleman here, right, who one Friday afternoon, Eric Pierce and I, one of my colleagues, just decided we're going to go over and just film what we do because, we give anesthesia all the time. We don't really pay attention, all right? So I just wanted to show you. There's some very obvious things that you can just glean from this little video. So this is a gentleman. I'm going to have him track my finger for a second here.
Let me just check one other thing here. What I'm going to ask you to do is, so I'm just going to
turn his head.
Let me turn it, okay? You don't have to, I'm going to turn, yeah, there we go. I'm going to turn your head.
All right, so that's the oculocephalic reflex. I turn your head this way, your eyes go the other way, and vice versa. All right, so it's totally intact. There's nothing abnormal there, all right?
Two more times.
And I'm going to have him pull on my arm here, pull on my hand. He's got normal motor strength. We bond with our patients before we start. All right? Okay. All right. So, and now, if you've never seen this before, what we do is we have the patients breathe oxygen. We have them breathe 100% oxygen for a period to fill their lungs up just in case there's a problem. We've created a margin of safety. So that's what's happening now. Okay, so right here, where the words come on the screen, right here, we've given him an injection, a standard dose of propofol. Now I'm going to ask him to track my finger, so just watch this.
You tell me if you can no longer do it. It's fantastic. Just keep doing it. Just keep doing it. That's great. If you can no longer do it, just tell me.
Got to ask,
Sir.
That's for effect. I've seen this once before. All right. So right now, she's got to breathe for him because he's basically unconscious, all right? So watch what happens here. All right, turn his head now, look. His eyes are fixed in the midline, okay? He's not breathing, he's unconscious. His heart's still beating, the beep, beep, beep. He's still with us, all right? All right. Now watch his arm, though. Watch what happens here. Boom. All right, it's like spaghetti, okay? So what just happened to him? So this is why, saying we don't understand how anesthesia works really doesn't make a lot of sense, because I can tell you very quickly what happened to him. You can relate it to what you just saw clinically. When blood enters the brain, sort of the brainstem down here, it comes up through what's called the basal artery, which runs right along here. The first thing it hits are the respiratory centers, and it basically inactivates them, all right? Right on the road, all right? Then you come up to this point right in here where all the arousal centers, remember consciousness has two components. It has arousal, the brain has to be activated, then it has cognition. The cortex has to process. So basically what happens is you take out these arousal centers. How do you know you took them out? Well, the nerves that control the eye movements are sitting right here. So when the eye movements go away, you've basically taken out those arousal centers, all right? And then the final thing, like why does he become flaccid? Well, I put this guy over here because this is something that NASA's interested in. Because, when the astronauts come back from the space mission, you never see them hop out of the space and go, hey, that was one hell of a good ride, I had a good time, sort of thing. Because they're like spaghetti, right? They can't move. You actually carry them out. You see them 2 days later for a press conference. So one of the things that happens is that we lose motor control, and it's these relay nuclei here which are important, or play an important role of that. So we just saw all those things taken out right there, those three things. So stopping breathing, losing muscle tone, and becoming unconscious. Now, so how can we track this, like in the operating room? So what happened to his brain? So if you look at the EEG, when he was unconscious, this isn't his EEG, this is one of my patients I was taking care of in the operating room. You have these oscillations like this. There's a slow oscillation, and there's a faster oscillation, which is about a combination of the slow oscillation, which is about 1 cycle per second here, and a combination of 1 cycle per second and 10 cycles per second. All right, so this is the alpha oscillation, which is this one here, and this is the slow oscillation. So those two are there, all right? Now, propofol acts at GABA A receptors. it basically induces inhibition. Now, the key thing is the following. This is something that engineers can really appreciate. The system doesn't shut down because there's so many connections and it has so many degrees of freedom, it actually starts to oscillate. That's why you see these oscillations, all right? So you apply a stimulus which shuts the brain down, and it just makes them go, it just finds these lower modes. And these are in some sense, kind of preferred modes that the system finds, all right? So here's what we figured out from our research. So the propofol hits these areas up here in the cortex, right? It generates patterns like this in the operating room, this alpha oscillation here, the slow oscillation. And now what we figured out from our studies is that this alpha oscillation here is probably a resonance between the cortex and the thalamus. So it's like this. So here's the hum, right? So you say, okay, you're giving me this lecture now. Okay, what I want you to do is give me the same lecture, and I'm going to give you one frequency, and you get to keep the amplitude the same. So what I'm saying now becomes hmm. So that's effectively what's happening here when you see this appear on the EEG. Right? Now, and the other thing that we know about, the reason we know this is because when we've measured EEG simultaneously across patients, you have this highly coherent activity across the front of the head. We've also just verified this in animals. So you have this oscillation which is going back and forth between the cortex and the thalamus, which makes it difficult for you to transmit information. Right? Then the slow oscillations, what happens there is that when the slow oscillations come on, and these are actually slow oscillations measured in patients that have electrodes in their brains because they've come for epilepsy surgery to locate the foci, the epileptic foci. So they need anesthesia, so we record during the induction of anesthesia, and we see these slow oscillations appear. And when these slow oscillations appear, what we've learned is that the areas of the brain are no longer coordinated. And in fact, the disruption can be measured by the degree of sort of lack of coordination among the slow oscillation cycles. And then the neural activity, this is a representation of neural activity. The neural activity here, right, is governed by this slow oscillation, whereas the neural activity over here would be governed by this slow oscillation. So if you have different locations which are not in sync, it's more difficult for them to communicate. Right? So who cares? All right? So here's the deal. So if you're 30, right, and you're under anesthesia with propofol, your EEG looks something like this, all right? There's a 10 hertz oscillation, there's a slow oscillation. You get a little older, looks like that. Not bad, kind of the same. A little older, 81, looks like that. Same same drug, administered to the same patient. If you were 56 though, right? These guys are the same age, but look at their patterns, right? So what's interesting is you can actually see differences in what might be brain age under anesthesia. After I saw it, I said, guys, you're not putting that EEG on me anymore. We don't need to know anything else, all right? The 3-year-old looks like this, and the 14-year-old looks like that, all right? So forget about anesthesia for the purpose of anesthetizing someone, right? This is kind of an experiment. It's a natural experiment, right? All we've done is we said, okay, we've given you the drug, and what we're doing is we're measuring your response. And the response is very, very different across age and probably across what is really relevant, brain age, all right? All right. So a couple of years ago, the New York Times did this little piece on our laboratory, and this was one of the letters to the editor. This person wrote in and said, I had my first corrective surgery for infrared paralysis, that's polio, in 49. The bill for anesthesiologist was $400. My mother was horrified, but my father calmed her when he said it was $50, for the procedure, and $350 because he knew how to wake her up, all right? Remember, as I tell people, any fool can knock you out, right? In anesthesiologist to bring you back, all right? So one of the things we haven't focused on in anesthesia is to actively bring back people from surgery. We actively turn the brain off, but we don't actively turn it back on. So my colleague Ken Salt has been working on this. So, let's see. He certainly has been working on it. Here. Ken has another video over here, which I just pulled up. All right, he's been working on it. So this is one of our rat volunteers. All right, and he has an electrode in his brain, in his ventral tegmental area. All right. And I'm going to put this little pointer like right here, okay? And he's getting anesthesia. He has an IV in his tail vein. Okay, and you can see he's breathing. There's the IV in the tail vein. He's getting propofol continuously, all right? And the microstimulation is off, all right? Now, when you turn the stimulation on, watch what happens. See how he starts breathing right away? Now watch this. Remember, he's still receiving anesthesia. He's anesthetized. And he goes, Ken, I'm out of here. All right. So what just happened there? All right. So I'm not sure why these aren't projecting. Let's see. All right. So I'll just tell you what happened since this doesn't want to. So what we did there was the following. We inserted electrode in his ventral tegmental area. And this is an area which is responsible for releasing dopamine. Not dopamine for the purpose of movement, which is substantia nigra. That's the area that's deficient when you have Parkinson's disease. But dopamine, which is important for cognition, right? And so one of the things we've also done, the first way we established this was not by going in and doing an invasive stimulation, but actually giving the animals Ritalin, right? And it turns out that if you give intravenous Ritalin, you can wake the animals up from anesthesia the same way that we did with the stimulation. So why should this matter? Well, one of the things we're concerned about tremendously after anesthesia is this loss of cognitive function, that is particularly in older people. And so one of the things we're looking at is maybe we should turn the brain back on actively. And you can sort of see that that's indeed possible. So if you look at this here, you've seen like the New Yorker's view of the United States. So this is the anesthesiologist's view of like neuroscience, right? Here we are like in the center, right? But it's actually true, I can create a locked-in state in patients very easily. It's very simple to do. Coma recovery is very much like recovery from anesthesia. We've studied tons of parallels. Ketamine is actually being used to treat patients rapidly for chronic depression. A Parkinsonian syndrome is generated if you give people high doses of opioids very quickly. We saw some breathing control there. We actually have drugs in anesthesia which actually produce states which are very much like sleep. And ketamine is also used to stimulate schizophrenia. All right. So what's the deal? Well, this is an area of science which isn't viewed as neuroscience, but yet it could play a central role in helping us to unravel a large set of things which relate to brain function. So I'll stop there. Thank you.
Phil, you got to go. Okay, thank you so much. I'll apologize for you. Take care. Oh, thank you. Phil's leaving now. I'll just take that. Yeah, that's fine. Phil's going now. Oh, great, thank you. So
Emory, follow.
if Emory has to go, that's fine. So you guys should go. You sure? Okay, so you're going to go. As is actually a rule for anyone that knows academics, whenever we get a chance to talk about our own work, we're very excited and we tend to run a little longer than maybe we intended. So we have some time to answer the questions, but Dr. Rubin had to leave to be at another meeting, so he apologizes. He said, I'll take care of all of his questions, so I'll have the authority to speak for the White House now, I guess. So I'll enjoy that. But anything that appears on the front page of the New York Times, I'm not responsible for. So we heard a lot of very interesting different topics, and hopefully you saw some of the broad range of impact in terms of the way this kind of basic research can ultimately influence our lives and influence the way technology and society shape our lives. I have a couple questions for people, and then we'll take some questions that were handed to me. One of the first ones, I want to ask the whole panel, but I want to ask Emory first because he's not from Carnegie Mellon, although he knows Carnegie Mellon well. And so maybe he can give a sort of 10,000-foot up view. Is in what ways does, obviously every university has strengths and weaknesses, and so in what ways is Carnegie Mellon very well poised, and Pittsburgh more generally well poised to participate in this kind of effort? And where are the things that we should be focusing and thinking on beyond what you heard today and also with what you heard today?
Oh, I think you have enormous strengths in a number of areas. I think that, you know, Tom hit on a main one. That is, you have this incredible expertise in computation, so machine learning, computer science, as well as statistics. And the two groups are already working together. Then you have the CNBC, which acts as a very important integrative force across sort of neuroscience as well as, you know, computation. And those are two key building blocks. I mean, I had the good fortune to be on the Brain Initiative working group, and that's one of the things we talked about. They want to see efforts across different disciplines because that's where the answers are going to be found. And in addition to that, when you add the other leg of what is your CNBC, you bring in the University of Pittsburgh and you have, you know, the medical school. I mean, you really, I think, have all three legs that are essential for making, you know, phenomenal progress in this area.
Anyone want to add anything?
I would just say a little bit about, I think beyond what we've seen here today, I think there's another strength of Carnegie Mellon is in technology development more generally. And so when it comes to applying new technologies that come from engineering or probes and reagents that come from our biologists and our chemists, I think that that's another strength where we can now have tools that allow us to see things that we couldn't see before. And I think that's an additional strength beyond what we mentioned here.
So that actually leads exactly into a question that several people raised, which is, several people raised the issue of Kurzweil's idea of Xeroxing the entire brain. But then just more generally, how do you build a model of the brain at the detail that the Brain Initiative seems to be asking for? Is that one possible? And what are the technologies and ideas that we might be able to use to attack that? So.
Well, I mean, I guess I'll start off. I mean, I think there are two questions to me that are related. One is, how can you see the things that you need to see in order to take a picture or take a movie of what is going on in the brain? And I think there are a variety of different scales and levels at which this can be approached. I think in the early discussions of the Brain Initiative, there was this idea of trying to record every action potential from every neuron, which I think is, you know, it's science fiction certainly at this point and maybe forever. But I think we need to be able to do more and more to record at a finer grained level as well as at the sort of coarse grained level of whole brain. And then we need to be able to put that into the right context, put that into a theory that allows us to relate why it is the behavior of a neuron, altering the behavior of a neuron or a population or a type of neurons will have the consequences that it does at the level of behavior. And I think that's a critical component that will require us being able to see more, build better models, and actually also, in a quantitative way, understand what's going on at the level of behavior.
One quick comment. I just want to put this a little bit in perspective. You have to realize that all of the work that we heard about today that involved brain imaging would have been impossible 20 years ago. When I was an undergraduate, I was interested in intelligence. I thought maybe I should major in psychology, but I realized how futile it was. They didn't even have an oscilloscope. So in 1990...
Oh, we had oscilloscopes.
I mean, this kind of oscilloscope. In 1995, a miracle occurred, really, the advent of widespread use of fMRI and brain imaging. So just to put this a little bit in perspective in terms of where we are today, as I think I would agree with Nathan, we're pretty far away from being able to record every neuron firing. But really, we went from 0 to 65 in 1995, and it's been accelerating since then. So right now, we're roughly 15 years in. This is like 15 years after the invention of the telescope, when Galileo said, gee, maybe we should look at the stars and see what's up there. We're 15 years in. It's roughly comparable.
Marlene, you had something you wanted to...
Yeah, so I just wanted to add just another piece to the puzzle, and that is that even if we were able to record the action potentials from every neuron, and even if we had available to us the most amazing oscilloscopes that humans could ever invent, the question is whether that would suffice to allow us to understand the neural basis of behavior. And I would argue that the answer to that is no, because we, on the other side, still need to have very detailed characterization of behavior. We need to be able to understand the behavioral end as well. And so psychologists need to play a critical role in this domain in order to be able to talk about the complex and abstract behaviors and the richness of the behavioral repertoire that humans are capable of.
Got a lot of questions here. One of the other questions I think that emerged from several different people was, how do some of the things you've talked about relate directly back to sort of both communication of science? How do we communicate? Because information that we learn is only as valuable, of course, as if it is valuable to the public. And Phil touched on that a little bit, but I think people are very concerned about how understanding communication. And then also, how do you translate that more into clinical practice? So Marlene, you want to lead with that?
It is obvious that one of the ultimate goals of this pursuit is to be able to impact people's lives. And as we already heard in great detail, the number of people who suffer from various kinds of brain and mental disorders is really enormous. It behooves us as a society to be cognizant of this and to work very hard to ameliorate the difficulties that these people experience. And so the question is, how do we take this on? I would argue that we take it on carefully, rigorously, step by step, but we can't wait until all the knowledge has been accumulated. We have to begin right now to implement what we already know, to design and refine the kinds of intervention and rehabilitation procedures so as to begin to have impact as soon as possible.
Yeah, I think there's another part of it too, and it was suggested a little bit by what I was showing you. There's a tremendous amount that we can learn from patients, and I think that we really need to, there's a huge technology push on, but I think the observations of, people's behaviors, as Marlene was talking about, but also just like the simple observations I was showing you of this patient, in the video there, and actually trying to really relate that directly to what goes on in the brain is something which we have to make a directed effort to do. I mean, we were very fortunate in those experiments because there is an experiment of nature. People come to have epilepsy surgery. They have electrodes in their brains. So it's then getting the permission to then go ahead and use that information to gain new insights, because that's a rare opportunity to see what neurons in the human brain do, not just what the neurons in rat brains or let's say in monkey brains, what in the human brains do. And I think that's a tremendous situation. There's more and more of that, and we should enable that type of research, I think, as much as we possibly can.
We've got to finish up now. I had requested that all the panelists come up with a 1 to 2 line, 15-second summary of how the brain initiative might impact or relate to what they're thinking about. So I'm going to ask, just go down the line, we'll do that. And I think some of us can stay afterwards to answer more of the questions you asked if anyone wants to come out and talk. So Tom, why don't you lead?
Well, I'll just say that I expect the brain initiative will cause a revolution in the field of our understanding of the brain. My hope is that it also causes a corollary revolution in the field of artificial intelligence, and that these two fields talk a lot more to each other in the future.
So the thing that Mike didn't say about his instructions was that he asked us to try and be funny. And so the thing that I would like to say is, if the brain initiative could achieve one thing, I would like it to be able to achieve a transplant of Emery's sense of humor into me.
It's really hard to follow that one.
That's where I didn't even.
I'll just say that I'm greatly encouraged by the quality of the science, as shown by my colleagues on the panel, and also by the energy in this domain. I really am of the opinion that we're on the threshold of a new era, and that the collaborative efforts of CMU and its partnership with the University of Pittsburgh can contribute significantly to this new domain.
So I don't know what I can say after Nathan. Just, as you can see, I'm blushing already. But I think that what I said earlier is really the case. I think one of the things that we emphasized in the initiative was trying to move efforts forward to try to learn as much as possible from humans, because that's where we want to have the ultimate impact. And I think, therein lies, the challenge, and therein lies the exciting opportunity to bring technologies that are being developed, directly into humans. I'll just make one very quick point. The brain stimulators that are used right now for Parkinson's disease are really based on a 1980s design for heart stimulators, right? We've made a lot of progress since then. But those things, because of various issues, haven't made them into human care. So I think there are enormous opportunities there. Thanks.
Thank you for coming.